photosynthesis, and well done for starting early. Hope some of you managed to go to the socrative.com and carry out some of the quizzes. Put on a 18 question, 16 question multiple choice quiz. Should have enabled you to start thinking about photosynthesis. That's running, and it should be giving you feedback on your answers. And I'll leave it running after this webinar, so if you haven't had a chance to do it, then you will have a chance to do it afterwards. I'll leave it running till tomorrow morning. Next webinar next week is on gas exchange in fish, mammals and insects. If ever you have any questions, then the Q&A box at the top of your screen, if you just type a question into that, then I should be able to answer it live. If ever you want me to go over something again, just say that and I'll go over it. It's a lovely day, and thank you for joining me, and I hope you find this useful. So what we're going to cover is we're going to cover photosynthesis. And remember, we start with the concept that almost all plants are photosynthetic autotrophs, and some bacteria and protoctistans. Now remember, with a cyanobacteria don't have chloroplasts. In fact, chloroplasts were originally free-living bacteria incorporated within larger cells, and those larger cells are eukaryotic. Now, the prokaryotes can photosynthesize, so those are the cyanobacteria, and also the protoctistans, so things like euglena and seaweed. And then we're more familiar with the higher plants and the mosses and the flowering plants, all of which photosynthesize. And photosynthesis is about generating your own organic matter through capturing light. And the own organic matter they're referring to here is carbon-hydrogen bonds. Once you take carbon dioxide and you join carbon dioxide with water, then you are effectively producing carbon-hydrogen bonds, which can then be used to make glucose, cellulose, proteins, fats, DNA, more plant. Remember, those bits of more plant are then consumed by us, which are heterotrophs, and they then used to power all heterotrophs. So every piece of carbon in you was originally carbon dioxide in the air, and that was then taken by a plant, used in photosynthesis, used to make more plant. So all of your carbon, all of your nitrogen, all of your amino acids, DNA, RNA and fats all come from plants. Now, sunlight is used to change the chemical energy, to change the carbon dioxide into carbon-hydrogen bonds. So this is the big picture. We take water and we take light, and we use the water and light to produce ATP and NADPH. The water is split, giving you hydrogen ions and, crucially, electrons. And the electrons are used to reduce the NADP. The ATP is made by chemiosmosis, which is the flow of hydrogens across a membrane, generating ATP when those hydrogens flow out through ATP synthase. The ATP and the NADPH are then used to reduce carbon to produce, it says sugars here, but it's carbon-hydrogen bonds that are then used to produce everything in all living things. So the big overview is the light reactions produce ATP and NADPH, and then the Calvin cycle takes the ATP and takes the NADPH and enables you to reduce the carbon dioxide to carbon-hydrogen bonds. The ATP that you generate is a way of transferring the light energy to giving you the energy for synthesizing the sugars in the Calvin cycle. So we're going to cover today how you absorb light, the structure of chloroplasts, light-dependent reactions, light-independent reactions, and a little bit on limiting factors. Okay, so we start off with why are plants green? The reason they're green is that visible light covers a range of wavelengths between 4 and 750 nanometers. The reason plants are green is because they don't absorb the green wavelengths. So you can see a large number of different colors because you can perceive different wavelengths of light. Plants look green because they reflect green light and they absorb the blue and the red wavelengths most strongly. Now, within a plant cell, we have chloroplasts, and chloroplasts are bright green because they are absorbing the red and the blue wavelengths. It is the thylakoid membrane within the chloroplast that has in it the photosynthetic pigments. And there are a number of different photosynthetic pigments which absorb a range of wavelengths of light. But the wavelength of light that they don't absorb is that which you perceive as being green. Now, we have a concept of absorption and action spectrum. Now, make this go up to the top. Here we go. Now, absorption spectrum is the range of wavelengths which are absorbed by individual pigments. Now, students find this a bit of a challenge sometimes. So if I draw you a couple of pictures to explain what's going on here. So if we have a range of wavelengths of light and we took chlorophyll A and we put chlorophyll A in a cuvette and then we shine a wavelength of light through the cuvette and we've put chlorophyll A in here. Now if we shine, say, 300 nanometer light, so we have wavelength of light at the bottom and then we have percentage absorption, then we could measure how much of the light gets through to the detector on the other side. If we then increase the wavelength, so we change the wavelength to say 400, it might absorb a little bit more, to 450, to 500, and so on. So when we produce an absorption spectrum for chlorophyll A, we change the wavelengths of light that we shine through chlorophyll A, and then we measure how much of that light gets through. And we produce a spectrum, and when we join it up, it looks a bit like this. So we see that two wavelengths of light in particular are absorbed very strongly. Now, if you repeated this exercise with chlorophyll B, so we put chlorophyll B in here, and we shine the same wavelengths of light through, we see a slightly different absorption spectrum, because chlorophyll B absorbs slightly different wavelengths of light. If we repeat the exercise for some of the accessory pigments, such as carotenoids, again, we get a different range of wavelengths. So when we see an absorption spectrum, we're changing the wavelength of light, and then we're seeing how much of that light gets absorbed by each particular pigment. So if we shine violet or blue, we can see that there is a wavelength strong absorption by the chlorophyll A, whereas if we shine orange and red, there's also a strong absorption. Whereas if we shine green and yellow, there's absorption of low levels of that light. Now, often you overlay onto this graph an action spectra. Now, an action spectra is achieved in exactly the same way, but instead of measuring the absorption, you measure the amount of photosynthesis that is caused by each different wavelength. So of a percentage of the maximum amount of photosynthesis that could occur, when you shine the different wavelengths of light through an intact plant, and the intact plant you put in here, and then you shine different wavelengths of light by using a filter, and then you measure the percentage of the maximum amount of photosynthesis that could occur. And then you change the wavelength of light and you plot a point for each different wavelength of light. And what you see when you do that is you get what's called an action spectra, which is the action of photosynthesis. And remember, you're dealing with an intact plant here, so that the action spectrum reflects the absorption of lots of different wavelengths of light by all of the different pigments that make up the photosynthetic pigments that are in the chloroplasts. So the action spectrum is the resultant of the absorption of light by the different pigments in the chloroplasts. Okay, so chloroplasts contain several different pigments: chlorophyll A, chlorophyll B, carotenoids, two types of carotenoids, which are carotenes and xanthophylls. Now, these are the carotenes and xanthophylls, which you see when in autumn, when the leaves turn yellow and red, and that's because the plants are reabsorbing the chlorophyll A and chlorophyll B from the leaves because they contain magnesium, which is an important resource. So the advantage to a leaf of having more than one photosynthetic pigment is that it's able to absorb a larger range of wavelengths of light, therefore can channel more energy for photosynthesis. So here's chlorophyll A. We've got a magnesium ion in the center of a porphyrin ring, and this has a delocalized electron which is going to be displaced by the absorption of sufficient amounts of light energy. Now, the difference between chlorophyll A and chlorophyll B is chlorophyll A has got a methyl group here, and it's replaced by a carbonyl group in chlorophyll B. Now, as a result of that, they absorb slightly different wavelengths of light. They have a phytol tail, and this is a non-polar tail, which is there to stick it into membranes. Quite a common question is to look at the RF values of, if you take essence of leaf and grind up essence of leaf, and then you put the leaf pigment here, and then you put it into a solvent and you carry out chromatography. Now, chromatography means that the solvent is going to rise, and as it does so, it's going to move the different constituent pigments of the ground up essence of leaf, and they're going to separate into their different pigments. So what you do then is work out the RF, or relative front value, which is the distance moved by the spot divided by the distance moved by the solvent. And this gives you an RF, or relative front, because it's the distance moved by the spot relative to the front, which is the solvent front, this bit here. Now, this came up in a recent OCR question, which says 4 different pigments were extracted from a plant and thin layer chromatography was carried out. And using the figure, what can you conclude about the composition of the pigments? Well, what they're looking for you to say here, the pigments are here, so the pigments have been placed there, and then the pigments have been spread out. So what you can conclude is that A is made of 2, B pigment is made of 1, C is made of 3, and that D is made of 1. What you can also conclude is that the same pigment is present in B and C there, in A and C there, and then all of them except for B contain this pigment. And that comes up quite often. Okay, I've got a few questions. What's the function of the magnesium in the porphyrin ring? Do you know if we get the RF formula in the exam? The function of the magnesium in the porphyrin ring is that there is an electron in that ring, and the magnesium is absorbing the light. And do you know if you get the RF formula in the exam? Normally they expect you to know it, that its relative front, which is distance moved by the spot divided by distance moved by the solvent. Okay, moving on. So this is the connection between absorption and action spectra. Remember that the action spectra is the resultant of the absorption spectra, and the absorption spectra depends on lots of different wavelengths of light, and then a point showing how much of that light is absorbed by each individual pigment. And that when that light energy gets absorbed, that creates the action spectra, because the light energy is then used to carry out photosynthesis. So action spectra is a graph showing the degree to which different wavelengths of light affect photosynthesis. Obviously, action spectra is closely correlated with the absorption spectra, and that shows that the main harvesting pigments are chlorophyll A and chlorophyll B, and the carotenoids. Okay, structure and function of chloroplasts and where they are. So if you're looking at a standard mesophyte leaf, then you've got the chloroplasts are mainly in the palisade mesophyll and also in the spongy mesophyll. And remember, you also get chloroplasts in the guard cells in the lower epidermis. Chloroplasts are here in the mesophyll cell, and then this is the structure of an individual chloroplast, and then this is one taken with an electron micrograph. It's really important you do know the structure of a chloroplast, and also you know the functions of each different part of the chloroplast. So the chloroplast contains stroma, which is the cytoplasm of the chloroplast. In fact, was the cytoplasm of the prokaryotic cell that formed chloroplasts all those billions of years ago. It's got grana, which are stacks of thylakoids, and the thylakoids are the membranes within the chloroplast that contain the photosynthetic pigments, that contain the chlorophyll. Now, it's important also that you know what the different parts of the chloroplast do and why they're there, and what the connection is between structure and function. So remember, the chloroplast isn't a sphere, it's a squashed oblate spheroid. And as a result of that, it's got an increase of surface area for the absorption of carbon dioxide, because carbon dioxide is going to be absorbed, because it's going to be diffusing into the chloroplast from the cytoplasm of the mesophyll cell. And the reason it's diffusing in is it's going to be being used, because remember, the carbon dioxide will be joining with a 5-carbon compound catalyzed by rubisco to turn it into glycerate-free phosphate and then triose phosphate. So things that are important here is that by stacking the thylakoids into grana, and the grana is the stack of thylakoids, by doing that, you're maximizing the area for light absorption. You have ribosomes in the stroma, and those ribosomes carry out protein synthesis. The chloroplast has also got a loop of DNA, which contains the genetic code for the proteins of the Calvin cycle, which will be made by the ribosomes. You've got starch grains and lipid droplets. Now, the importance of starch grains and lipid droplets is that the chloroplast is going to be producing soluble sugars. Now, if you produce soluble sugars, you'll lower the water potential of the stroma. And if you do that, water will move in. Now, because this is an organelle, not a plant cell, you won't be able to have a cellulose cell wall around the outside, which will restrict the expansion of the stroma, so that then there could be consequences to the influx of water. So that's why you would want to store the sucrose as starch or as lipids. Okay, Talking about the relationship between chloroplast structure and function. So remember, the palisade cells are here. High density of chloroplasts, and they're close to the top of the surface of the leaves because the light's going to be shining like this. Remember, the carbon dioxide is being absorbed in the spongy mesophyll and also in the palisade mesophyll because there are gaps between palisade mesophyll cells. More links between structure and function. So thylakoids give you a large surface area. The chlorophyll pigment molecules are embedded in the thylakoid membrane, which forms the photosystems, which we're going to talk about in a minute. The thylakoid spaces are quite low volume, and that's important because it enables a high hydrogen ion concentration. Because remember, the hydrogen ions are being pumped into the thylakoid spaces by the movement of electrons along the thylakoid membranes. And those hydrogen ions are then flowing back out through ATP synthase, which is generating ATP. And ATP synthase is also embedded in the thylakoid membrane. Now, the stroma contains all of the enzymes involved in the Calvin cycle. And obviously, the most important enzyme here is rubisco, which is RUBP carboxylase, which carboxylates RUBP. Does exactly what it says on the tin. Okay, ploughing on. So similarities and differences between mitochondria and chloroplasts comes up quite a lot. So things that you've got to consider here is that they both have an outer membrane, which keeps them separate from the cell cytoplasm. That they both have thylakoid membranes and an inner membrane, and that those membranes are where the electron transport chain occurs. So on the inner mitochondrial membrane, you've got the electron transport chain, which moves hydrogen ions across, and that's driven by NADPH, and that obviously the electrons end up on oxygen producing water. And that gives you the hydrogen ion gradient for chemiosmosis. And it's exactly the same in the thylakoid membrane, that the flow of electrons along the thylakoid membrane gives you the hydrogen ion gradient so that they then flow out through ATP synthase. Mitochondria have folded or invaginated membranes forming cristae, which gives you a large surface area for reactions to occur on. And the same is true of the stacks of thylakoids forming grana. They both have a cytoplasm. One is called a stroma, the other one's called a matrix. And in both of those is where the enzyme-controlled reactions occur. So it's the Krebs cycle that occurs in the matrix, and in the stroma, it's the Calvin cycle. Okay, so we start with the light-dependent reactions. So light-dependent reactions carry out photolysis. So lysis is to split, and photo is using light to do it. So the photolysis of water involves taking water and then splitting it into oxygen, which is a waste product, electrons, and hydrogen ions. I'm going to talk about that a lot in a minute. The electrons are used to reduce the NADP to NADPH, and the flow out of the hydrogen ions is used to phosphorylate ADP to ATP. Now, the light-independent reactions occur in the stroma, and this involves taking carbon dioxide and reducing the carbon dioxide to form carbon-hydrogen bonds. And that requires the electrons being carried by the NADP, which are then oxidized from NADPH to NADP, and this makes triose phosphate. And at the same time, you're consuming the ATP, turning it into ADP. Another question that's come up recently in the OCR sample papers looked at phospholipids. So phospholipids play a crucial role within plant cells. Which of the following statements linked to the importance of membranes is or are true? And they say that phospholipid bilayers within the chloroplasts are impermeable to protons, Remember, that's very important because you've got to build up a hydrogen ion gradient so that those hydrogen ions can then flow out through ATP synthase, that they contain electron transport chain proteins. Again, that's important because that's how you move the hydrogen ions from the stroma into the thylakoid interior. And then finally, well, statement 1, ATP synthase embedded in the thylakoid membranes maintains the chemiosmotic gradient. Well, that's not true. So these two are true. So this gives us C because it's only 1 and 3. Okay, moving on. Concept of a photosystem. So a photosystem is a lot of different pigment molecules, and these all absorb different wavelengths of light. So different wavelengths of light are absorbed by these pigment molecules, and that energy is then transferred through these pigment molecules to a single chlorophyll A molecule. And this single chlorophyll A molecule is known as the reaction center. So now we're going to talk about the light-dependent reactions. So I'll draw you a few pictures first, and then we'll look at the PowerPoint. So concepts with the light-dependent reaction is we have a thylakoid membrane, and then we have another thylakoid membrane, and then we have stroma and stroma here. Now we start with a photosystem that we're going to call photosystem 1. And the photosystem 1 absorbs a large number of different wavelengths of light, all goes to a single chlorophyll A molecule, which is at the reaction center, and then that loses an electron. Now the electron returns back to photosystem 1, and as it does so, it goes through an electron carrier, and this electron carrier uses the energy in the electron to move hydrogen ions across the membrane. And it's moving the hydrogen ions from where there are few hydrogen ions to where there are lots of hydrogen ions. So it's moving them against their concentration gradient into the thylakoid interior, into the gap between the two membranes. Now, those then flow out through our world's most wonderful and amazing enzyme, which is ATP synthase. So the hydrogen ions flow out through ATP synthase, and that efflux of hydrogen ions down their concentration gradient phosphorylates an ADP and produces ATP. Now this is known as cyclic photophosphorylation. Cyclic because the electron is being cycled back round to photosystem 1, and photophosphorylation because you're using light to phosphorylate an ADP to an ATP. Now, the product of that is ATP. So that's interesting, but not amazing. What's amazing is the next step of evolution, which was to evolve another photosystem. And this photosystem is photosystem 2. So again, we've got a load of different pigment molecules absorbing different wavelengths of light, gets enough energy to lose an electron. The electron goes through a chain of electron carriers, and those electron carriers move hydrogen ions across the membrane. And the electron is being moved from photosystem 2 to photosystem 1. Now the difference here is that when it gets to photosystem 1, the electron that's being lost from photosystem 2 does something completely different, which is that it joins with a carrier molecule called NADP. And this reduces the carrier molecule to NADPH. Now that NADPH and the ATP then go off and power the Calvin cycle, so the light independent stages. Remember that we're now missing an electron from photosystem 2. And this is where the most amazing thing of photosystem 2 occurs, is that photosystem 2 evolved the ability to split water. So it takes water and it splits it into oxygen, which is released as a waste product, hydrogen ions, which add to the hydrogen ion gradient to generate the ATP, and electrons, which are used to replace the electron that's been lost from photosystem 2. Now, because the electron goes from photosystem 2 to photosystem 1 and then on to NADP to reduce it to NADPH, the electron is not being cycled, so this is known as non-cyclic photophosphorylation. And the electron that's being produced by photolysis is the electron that's then used to reduce carbon dioxide to produce the carbon-hydrogen bonds, which are then used to produce the glucose and the amino acids and the cellulose and the fats and all things in everything, in all autotrophs and everything that is eaten by an autotroph, eaten by a heterotroph. Incidentally, the evolution of photosystem 2 led to oxygen being in the atmosphere. And the presence of oxygen in the atmosphere meant that aerobic respiration was possible. And almost all the oxygen in the atmosphere comes from photosystem 2. So without photosystem 2, there's no aerobic respiration and there's no you. So we have a lot to be grateful to plants for. Okay. So going back to the PowerPoint. So we start with photosystem 2, and that absorbs light, and the light is used to raise the energy of the electrons in the chlorophyll, which is called photoactivation. The electron is moved to along an electron transport chain, and that causes a proton pump, which moves protons from the stroma into the thylakoid interior. The hydrogen ions accumulate in the thylakoid space, so that leads to a decrease in the pH in the thylakoid space. In order to replace the electron, you carry out photolysis. The hydrogen ions accumulate here. The hydrogen ions then flow back out through ATP synthase, and that phosphorylates the ADP to form the ATP. That's all caused by the flow of electrons, and they come from photosystem 2 and then photosystem 1. Now, once the electrons got to photosystem 1, then the electron that's come from photosystem 2 via the electron transport chain to photosystem 1, more energy gets absorbed by photosystem 1, and that energy is then used to reduce the NADP to the NADPH. And the hydrogen ions flow out through ATP synthase. Sometimes you see this as what's called the Z scheme, and the Z scheme represents the energy level and then the progress of the reaction. So energy level up here, and then progress of the reaction on the bottom. And what it shows you is light hitting photosystem 2, the electrons going up in energy level to an electron acceptor, and then falling in energy level as they go down the electron transport chain. And that fall in energy level is because the energy in the electrons is being used to move the hydrogen ions across the thylakoid membrane against their concentration gradient, so that requires energy. And then that energy is used to synthesize ATP by chemiosmosis. And then you see a raise of energy level from photosystem 2 to the primary acceptor, and then you see the joining of the electron to NADP to form NADPH. So this sort of graph. The confusing bit is that they say energies used to make ATP, and they don't make it clear that that's through chemiosmosis, so that the fall in energy is due to the movement of the hydrogen ions against their concentration gradient from the stroma into the thylakoid interior. Okay, there's another question. Why doesn't the hydrogen ions run out in the thylakoid space as it's constantly pumped out to the stroma to create ATP? Okay, so anonymous attendee says, why doesn't the hydrogen ions run out in the thylakoid space as it's constantly pumped out to the stroma to create ATP? So it's not pumped out to the stroma. It flows out to the stroma, and it's being pumped into the thylakoid space. So the flow of electrons along the thylakoid membrane moves the hydrogens from the stroma into the thylakoid space. And that flow of electrons is driven by light, by capturing the light energy. So the hydrogen ions, having moved into the thylakoid space, then flow out through ATP synthase, and then they're pumped back into the thylakoid space by the electron transport chain, which is powered by ATP, powered by light, sorry. Okay, moving on. So here again, just to make the point yet again of what's going on, we've got photosystem 2, we've got photosystem 1, absorption of light, electrons traveling from photosystem 2 to photosystem 1. That energy is used to move the hydrogen ions from the stroma into the thylakoid interior. They then flow out through ATP synthase. So this part is pumping because it's moving against the concentration gradient, whereas this part, it's flowing out. And at the end of it, we reduce NADP to NADPH. So then we go on to the light-independent reactions. So the light-independent reactions end up producing triose phosphate, which is then turned into glucose phosphate. Glucose phosphate is then made into starch or into cellulose, or can be used to make DNA or amino acids. Light-independent reactions take carbon dioxide and NADPH and then use the energy from ATP. Light-independent reactions occur in the stroma, and they're called the Calvin cycle. So, the process. You take RUBP, so this is RIB, RIB. So just like ribose or deoxyribose, there are 5 carbons. The B is bi, which means 2. So it's a 5-carbon compound with 2 phosphates, ribulose biphosphate. And because it ends in O's, just like glucose, sucrose, or fructose, you know it's a sugar. So you've got a 5-carbon sugar, ribulose biphosphate. You join a 1-carbon molecule to it, which is carbon dioxide. And that produces a 6-carbon compound with 2 phosphates. Now that only exists fleetingly on the surface of the enzyme. And the enzyme is called RUBP carboxylase because it carboxylates RUBP. So we take a 5-carbon compound, we join it to a 1-carbon compound, we produce a 6-carbon compound which splits instantaneously to give you 2 molecules of glycerate 3-phosphate. Now, it is crucial that you see that at no point have you used any ATP or any NADPH in this step. So the reduction of carbon dioxide doesn't require ATP or NADPH. What the ATP and the NADPH is used for is to regenerate the RUBP so that the cycle can continue. The ATP and the NADPH are used to take the glycerate 3-phosphate and then turn that into triose phosphate, most of which is then turned into RUBP. So do remember that the reduction of carbon dioxide doesn't require the products of the light-dependent reactions, What does require it is the regeneration of the IUBP. So we go from the glycerate 3-phosphate, we consume ATP that comes from the light-dependent reaction. We consume NADPH, which is turned into NADP, and the NADP goes back to the thylakoid membrane where it picks up more electrons, which have come from photosystem 2, to reduce the NADP to form more NADPH. Now, the consumption of ATP and NADPH produce triose phosphate. Now, that triose phosphate is turned into glucose phosphate and then into starch, or alternately, it's turned into IUBP. So the vast majority of the triose phosphate is used to make more IUBP. But 1/6 of the triose phosphate is used to make glucose phosphate. Now, the reason why it's 1/6 is because each time the cycle goes round, you're adding 1 carbon dioxide. So if this went round 6 times, then you should make 2 triose phosphates. If it went round 3 times, you'd make 1 triose phosphate. If you went round 6 times, you'd make 2 triose phosphates, extra ones. Now, students look at this and go, well, there's 5 carbon here, 1 carbon here, produces 2, 3 carbons, and I'm happy with this. And then we've got 2, 3 carbons here. I'm quite happy with this. And then those 2, 3 carbons get turned into a 5 carbon, but somehow you get a carbon dioxide, a triose phosphate coming off here. And they go, well, how can you lose 1 triose phosphate, but you've made a 5 carbon IUBP? The point is, don't consider the Calvin cycle as occurring just once. Consider it as occurring hundreds of thousands of times a second or a minute in the stroma of the chloroplast, and then consider that you're capturing 1 carbon dioxide each time. So if it went round 3 times, you'd make 1 additional triose phosphate. If it went round 6 times, you'd make 1 additional glucose. It's just that every time the cycle goes round, you're capturing a single carbon. Okay, I hope that made sense. So here's another picture making the points I've just made. So carbon dioxide joins with IUBP, catalyzed by IUBP carboxylase, which carboxylates IUBP, produces a 6-carbon compound which splits instantaneously to give you 2 molecules of glycerate 3-phosphate. This step doesn't use NADPH or ATP because they're used in the next step, which is to take glycerate 3-phosphate and turn it into triose phosphate. That triose phosphate is then used to make everything in the plant, and consequently everything that eats plants. You then produce RUMP, ribulose monophosphate, which is then phosphorylated by an ATP to give you ribulose biphosphate, and so the process continues. Remember that IUBP is the world's most common protein, and also that IUBP evolved when carbon dioxide levels were much, much higher than they are now. So when IUBP evolved, carbon dioxide levels were probably around 5 or 6%. Nowadays, as a result of IUBP, they are 0.04%. Or, sorry, 0.04%. Now, that's because the action of IUBP has led to trees growing, coal, oil, gas, the storage, the sequestration of carbon, because IUBP has led to carbon dioxide being turned into organic molecules in living things. And when those living things get fossilized, they get removed from the atmosphere. So all of the limestone that you see, all of the oil, all of the gas, all of the coal, all of this was produced by the action of IUBP carboxylase, which is, I think, an amazing thought. So a little bit on the exam question. So this again came from the OCR new spec. During the light independent stage, triphosphate is synthesized in the chloroplast of plant cells. State two possible uses for this molecule within the plant. So two possible uses might be to make amino acids, or starch, or cellulose, or DNA, or RNA, or to make more IUBP. And from which molecule is TP synthesized? It comes from glycerate 3-phosphate. So back to our big overview. Remember, the process involves taking light, using that light to split water. That occurs in the thylakoid membranes. The energy is used to generate ATP through chemiosmosis. The electron is used to reduce the NADP to NADPH. The NADPH is used to regenerate the RUBP because RUBP has joined with carbon dioxide to form glycerate 3-phosphate, and then that forms the triphosphate, which then forms the sugar, which is used for respiration, making starch, making amino acids, etc. Okay, having a look at some of the limiting factors of photosynthesis. So this is the effect of light intensity against the rate of photosynthesis. So light intensity is going to be the first thing that limits photosynthesis, because without light, you don't have any photosynthesis. When you increase light intensity, the rate of photosynthesis goes up. So if you got a graph that looked like this, and they asked you what was controlling photosynthesis, or what factor was controlling photosynthesis over this point, you would say that it was light intensity, because it's the factor that as you increase it, the rate of photosynthesis goes up. However, when it plateaus at this point here, it means that if you increase the light intensity falling on the plant, there is no increase in the rate of photosynthesis. And that's because the rate of photosynthesis is being limited by the Calvin cycle. The light, remember, is producing the ATP and the NADPH. The Calvin cycle is taking the carbon dioxide, and that's using it with the RUBP to produce the glycerate 3-phosphate, which then consumes the ATP and the NADPH. Now, when you're on this plateau here, it's carbon dioxide concentration, which is effectively substrate concentration of the enzyme, and it's also temperature, because temperature leads to the rubisco working faster because of kinetic energy effects. So when you get a graph that looks like this, over this part here, it's light intensity that's limiting the rate of photosynthesis, whereas this part here, it's some other factor that's limiting the rate of photosynthesis. So it could be carbon dioxide or temperature. Now remember, temperature affects photosynthesis because it's an enzyme-controlled reaction. If you had lots and lots and lots of light, so very bright light, and then you measured the effect of temperature on the rate of photosynthesis, then it's going to do this, which just reflects the effect of temperature on RUBP carboxylase, which past the optimum temperature, the enzyme begins to change shape because of the kinetic energy effects. Another look at a question. So plants respire and photosynthesize. Now, it's a common misconception that plants photosynthesize and animals respire. Remember, plants respire all of the time, otherwise they would be dead plants. So during the day, they respire. During the night, they respire. It's just that during the day, they also photosynthesize. Now, that may be obvious to many of you, but not so obvious to all students, somewhat depressingly. Now, with this graph, they give you the quantity of carbohydrate being produced by the plant and the use of the carbohydrate in respiration. So the dotted line is the rate of plant respiration and the consumption of carbohydrate, and then the non-dotted line, the continuous line, is the rate of photosynthesis. So they say explain the shape of the curve of the rate of photosynthesis. So explain would be that from 7 o'clock till 4:30, the light intensity increases, and then probably the temperature increases, so that you have more photosynthesis when there's light, and the more light there is, the more photosynthesis there is. So you get this increase here. Now, explain the shape of the curve for the rate of plant respiration. Well, at night, the plant is going to be respiring less because the temperature will be lower, because remember, respiration is an enzyme-controlled reaction, so that you'll get this increase during the day because the temperature will be increasing, so the plant will be respiring more. Now, what's happening at the points indicated by the letter L? Letter L is where respiration, which is producing carbon dioxide, is equal to photosynthesis, which is absorbing the carbon dioxide. So it's the rate of respiration is equal to the rate of photosynthesis. And that's often called the compensation point. Okay, another question. Will this webinar be sent to us to watch again? Not sure yet, thank you. But I'm having a think about that. Right, back to where I was. I've got a couple more points to make before that. I'll show you another couple of questions that tend to get asked. So if we had time along the bottom, and then concentration of RUBP and concentration of triose phosphate, no, concentration of G3P. Now, if at this point here, if you switched off the lights, what might happen? So, question from Karen saying, is the light independent reaction cyclic photophosphorylation? And the answer, Karen, is definitely not. So, light independent reaction is the Calvin cycle, and the Calvin cycle is about taking carbon dioxide and then adding it to the 5-carbon RUBP to produce 2 3-carbon glycerate phosphates that then get reduced by NADPH and the energy from ATP. The light-dependent reactions have cyclic photophosphorylation and non-cyclic photophosphorylation, which produce the ATP and the NADPH, which are then consumed in the light-independent reactions. So the light-dependent is cyclic and non-cyclic photophosphorylation, and the light-independent is the Calvin cycle, which consumes the NADPH and the ATP. Hope that answered your question, Karen. Another question. What is chlorosis, says Jay? Chlorosis is not having enough magnesium in the soil to make the chlorophyll. Okay, so back to this question I was trying to make a few points on. So if we gave you a graph of RUBP concentration and G3P concentration, and we switched the lights off here, what might happen? And the answer is that the level of G3P would rise and the level of RUBP would fall. Now, the reason for that is that the RUBP is being made by taking the G3P, which is then being turned into TP, which is then being turned back into RUBP. Now, the carbon dioxide is joining with the RUBP to form the glycerate 3-phosphate. So when there's no light, there's no NADPH and no ATP, but you can still have the reduction of carbon, so the level of G3P would go up. When you switch the lights back on again, so if we put the lights on here, then the G3P is going to be turned back into RUBP, and the RUBP levels will go up. Another point to make, whilst I'm still thinking about this, if a very common question or question that has come up, if you look at light intensity against rate of photosynthesis, then you get, let's do it a bit bigger, you're going to get this color. As you increase light intensity, the rate of photosynthesis goes up until it levels off when it's limited by the Calvin cycle. Now, the way you make the Calvin cycle go faster is you increase the things which make an enzyme-controlled reaction go faster. So you might want to increase the temperature to increase the kinetic energy effects, or you might increase the substrate concentration, which is the carbon dioxide concentration. Now, a common exam question would be a horticulturalist has a greenhouse and it is a bright sunny day in the winter, but it is cold. What should the horticulturalist do to his greenhouse to make sure that he has the maximum rate of photosynthesis? Now, the point they're making here is that you've got lots of light, but the rate of photosynthesis is being limited by temperature and carbon dioxide availability. Now, what the horticulturalist should do would be to heat his greenhouse. Now, the crucial point is how you heat the greenhouse, and it would be by burning a fossil fuel, because by burning a fossil fuel, such as having a paraffin stove in the greenhouse, you would both increase the carbon dioxide level and increase the temperature. So you would increase the factors that would make the Calvin cycle go faster. So if you had a cold day, but lots of light, then you're being limited by temperature and carbon dioxide availability, then burn a fossil fuel. Now, the counter one to this is it's a hot day, but it's very overcast and dark. What should the horticulturalist do? Well, the horticulturalist should open the windows in his greenhouse to cool it down, because the increased temperature will increase the rate of respiration, which means his plants will be respiring, but they won't have the light intensity which would cause the photosynthesis to occur at a sufficient rate. Right, another question. So why is carbon dioxide low on a cold day? It's not. I just didn't phrase that particularly well. So on a cold day, if it is bright and sunny, you have lots of light intensity, but you have a low temperature. If you heat the greenhouse, then temperature will not be limiting the rate, so that the rate will go up. If the temperature is high enough, then what's limiting it will be the availability of carbon dioxide. So if you increased both the temperature and the carbon dioxide in the greenhouse, you'd have a faster rate of photosynthesis. I hope that answered your question. I'm sorry if I gave you a confused question. Okay, that's kind of it on photosynthesis. If anybody has any questions, then I am more than happy to answer any of them on anything. But if certainly we could start with questions on photosynthesis, that would be great.